So good evening, everyone. This is the third lecture of this session and TA session, this third TA session. So I'm Shobik and I'm doing integrated PhD in Indian Institute of Science, Bangalore in biology. Uh, so in this TA courses, we will discuss any doubts regarding the courses, like whatever everyone like whatever sir has taught. Uh, I hope I hope I have, yeah, my mic is on. Yeah. So first I'll ask if there is any uh, doubt regarding the classes. There is only only one people have joined. So um, okay. So start the session. So there is no doubt. So first one note small that all whatever images I've used in this class was taken from the following books: the Harper's Illustrated Biochemistry and the Nature Principle of Biochemistry. I have uh, given one ebook in that drive folder in the course Excel sheet. So if you need anything, you can download those books from this folder and use them um, for reading. Okay. So this especially Leninger. So Harper's in things are like uh, written in a little gist format, and it's much like less detailed, is little detailed, but it's quite good for if you want to do small small like for the competitive exams or all. Leninger is the best book for this part, like for the biochemistry part. I would suggest you to read because it has very detailed discussions as well as quite detailed explanations, and many extra things are also written. So you can always go and read that. That would be best for understanding the course. Even I myself had read during my bachelor's or master's. I have also read Leninger only. Uh, and yeah, so go and read it. This is a like, classic book of the biochemistry. You can also consult White and White as well as Starr, but yeah, I preferred Leninger. So most of the things I will be sharing from either from Leninger or from Harper. Okay. So if you read Leninger, it will be very easy for you to answer many questions in your many of the competitive exams like JAM or GATE. Okay. Okay. So let's start. So first thing we'll learn about the nucleic acid. Like I'll just discuss briefly whatever has been taught, and they'll discuss some of the questions. Okay. Yeah. So as you know, nucleic acids. So there are total of there are total five. So in nucleic acid there are total five types of nucleotides. Correct. So there is like adenine, guanine, cytosine, thymidine, and uracil. Okay, and yeah, so you can see if you just and uracil and thymidine, there is just no reference. So there is only this methyl group is there. So if you demethylate or the thymidine, you get uracil. Okay, and yeah, um, so these are the nucleotides, and they are like they form phosphodiesterase. So when they are forming the bond, so these nucleotides, as you can see, so now when they attach to either they while forming the DNA, so okay, so nucleotides are these are so these are all the nitrogenous bases, right? So these are all nitrogenous bases, and you can see adenine. Now, so there are two types of nitrogenous bases, purine and pyrimidine. So as I told, like purine always base pairs with pyrimidine, correct? And so A will always have a double hydrogen bond with T or U, T in case of DNA, U in case of RNA, and G will always base pair with cytosine. And you can also see that A have a double bond so with T or U, and G has a triple bond with G or U. Okay, and so now if you delete this, right? Okay, sorry. Yeah. So now what to delete over here? Okay, and so now if I say any of the bases, if I uh, say any of this bases, for example, if I go for cytosine, so this is a nitrogenous base. Sorry. Uh, so cytosine is a nitrogenous base, and if I write things in here, so this is the site structure of not able to. 1 second. Yeah. So if I in this nitrogenous space, if I put a N-glycosidic bond with the adenine, for example, if I put it with cytosine, so how it will look like the cytosine in case of cytosine. So cytosine is basically C, then N, then double bonded C, NH2, CH, CH, O, NH, correct? And now if I yeah, no, so now if I say that this cytosine, so now if I remove this H and if I put a N-glycosidic bond here with a ribose sugar, like 5-carbon ribose sugar, correct, at this position here. So this whole thing will call it nucleoside, correct? And from in this nucleoside, if I just put H, OH, H, if it is a DNA, then it will be deoxy. So yeah, this is a DNA. So sorry, with the DNA, this will be deoxy. So HOH, and if I now attach a phosphate bond like this is the phosphate. So now in this phosphate, will in one side I will attach. So at its phosphate at 5 prime, it will attach to a DNA, and on the other end, at the end of this phosphate will be open at the 5 prime end. At the down, if you go, this OH will again it will like this CH2OH by this phosphate will get linked to this particular 3 prime OH, correct? So this will known this that we know as phosphodiester. So this is one ester bond, there is one ester bond, correct? And in between there is a phosphate, so phosphodiester, okay? And so if now in one side it is attached to a 3 prime OH, and one side it is attached to the 5 prime. Now, as always, at the 5 prime end of the carbon atom, the ibos carbon, at the 5 prime end, there will be 5 prime phosphate, 5 prime PO, yeah, 5 prime PO3 minus, correct? All right, say, yeah, PO3 minus 2. And at the 3 prime, there will be always a, with 3 prime, there will be always a 3 prime OH, correct? 3 prime OH. So yeah, so that will be always a 3 prime OH or a 5 prime phosphate all the time. So at the 3 prime end, it's 3 prime OH, and that's why the DNA has a polar nature of 5 prime to 3 prime. And DNA strands are always as an antiparallel, but this strand will go here, and this strand will go like this. Okay, this is a RNA, this is similar, but as you can see, the difference is as this 2 prime position. So as you can see, at the 2 prime, this is deoxy, at 2 prime, that is not deoxy. Okay, so RNA, ribonucleic acid. Yeah, and so when there is as you can see, there are this for example, in thymidine, as you can see, there are this NH and O that will form a double bond. This NH and O will form a double bond with adenine. With that, which one of this adenine? Now at this position, so this N as well as this NH2 should form a double bond, right? Adenine and thymidine, that's how it is forming a double bond. Why cytosine and guanine is always forming a single bond? Because you can see cytosine has 1, 2, 3. So one oxygen, 2 nitrogen this side. So then at the opposite side, guanine also you can see 1 nitrogen, another oxygen. There are 3, correct? So there are again, that's why it is forming 3 hydrogen bonds here. Okay. So there is one concept that, so most of the time, why is RNA, while we handle RNA, so RNA is very much prone to degradation. So what is, there are two reasons. One is like RNA is the RNA degradase enzyme, RNase A. This is a very stable enzyme. Very stable enzyme. Okay, RNase can be stable at quite higher temperature, and so it is very like abundant in the environment. For some reason, it's very abundant in the environment. Okay, it's very abundant in the environment. Okay, so one of the reason. Another reason is, so if you the RNA is very much RNA is very much so RNA is very much unstable, because if there is a OH ion, this OH can attack this hydrogen here, and since that this will like ultimately this H, then it will have a free ion. This will attack the phosphate bond, so forming a 2 prime. So it's a cyclic phosphate as well as this kind of like shortened ion. So that's why it gets cleaved, right? So this phosphodiester is blonde is getting cleaved. So that's why the alkali medium, the RNA is more unstable than DNA. How when DNA, since there is only will be H, will be no OH, because of, so this, so this cannot attack this hydrogen, right? Because this hydrogen is not polar, not a polar bond. So yeah, so DNA double helical structure, as you know, it has had been won Nobel Prize at 1962 by James Boston, Francis Cribb, and Maurice Wilkins. However, the Nobel Committee could not give to the actual person who who actually did the crystallization of our DNA, which was Rosalind Franklin. She unfortunately died of leukemia at quite young age. And as the lore goes, Maurice Wilkins, during showing she was unwell, Maurice Wilkins actually has stolen the data, stolen this particular data, like this DNA crystallography data from Rosalind Franklin, and shown it to James Watson and Francis Crick. So they actually then, after Rosalind Franklin did not survive, they actually went and solved the structure of DNA. And as you can see clearly that like from the structure you can say that there is like a cyclic structure, and there is this black spot here are arise from the recurring bases, so base stacking of the. So anyway, so always remember that the actually DNA structure who has been discovered actually by Rosalind Franklin, but unfortunately she could not survive to get a share of the Nobel Prize, because Nobel Prize is not given after your death. Yeah. So as they said that, so DNA runs in the anti-parallel way. DNA strands run in an anti-parallel way. So one strand goes from 5 prime to 3 prime, and the other strand will go from 3 prime to 5 prime, right? So like that, it's an anti-parallel way, and the distance between each stack, each of this base pair is around 3.4 angstrom, and the width of this DNA helix is around 20 angstrom. Okay, and there will be two groups, one major group where there will be more open structures like this, so major group, and there will be minor group where the cluster will be more closed, right? So you can think of like a those stairs if you see in old house, right? There will be like this there will stairs, right? So you can think of those stairs in the old house. There will be two parallel chain running like this. I mean like that. I'm not able to do it properly, but yeah. And yeah, and one turn of a DNA, approximately full turn of the DNA. So this is the one turn where, like, if you start from this, sorry, if you start from this particular position, if you go and again you start from this particular position and this particular direction, so this is called like one full turn. So one full turn of the DNA will contain one major group as well as one minor group. It's around 36 angstrom. Okay, and there are three different forms of DNA also. So A DNA, B DNA, and Z DNA. So A DNA is a right. Both A DNA and B DNA both have right-handed helix, but A DNA seems to be have a little shorter. And so the most common form of the DNA is, of course, the B form of the DNA, and A form of the DNA has 11 base pairs for each turn. However, uh, uh, in the B form of the DNA, there will be 10.5 base pair at each turn. Then there will be like the distance between the H bases, or the helix size per base, will be 3.4 angstrom. So this will be more open compared to ADN, will be more compact. Okay, and then there'll be sugar pucker will be 3 2 prime, and I will not go into details of this. There is also another thing is that there is a uh, left-handed DNA or zDNA. So zDNA is not very commonly found in the system, but recent studies says that they are found in some of the virus, and there are proper proteins that actually binds to this kind of zRNA. So they are like poly GC structure. The zDNA is known. So Z form is kind of still an active area of research. Yeah, okay. So now we'll come to the question of the question of today's uh, question of this week's, like some of the questions of this week 3. And so let's see. The only component in a nucleotide that has a net negative charge at physiological previous. Ribose. Ribose is sugar, right? Ribose or D-ribose or 2-deoxy-D-ribose. So ribose is a sugar like this, correct? It doesn't have any net negative charge, correct? So so ribose is just OH, OH. Just checking the ribose structure. Yeah, just yeah. So there will be H, H, and H, CH2OH, correct? So this two forms. So and if it is 2-deoxyribose, so deoxyribose will not have this, or which you will find in the DNA. Nitrogenous bases, of course, they are none of them. So phosphate. Phosphate is an ion, right? So PO4 -3, correct? So phosphate is an ion. So of course, this will have correct answer is the phosphate. The major force that stabilizes the double strand in aqueous solution is hydrogen bonding. Single DNS doesn't have this hydrogen bonding. They don't have any hydrophilic interactions. Ion interactions I don't is there, but stabilize the DNA because because that's why you need some salt or salt buffer to stabilize the DNA. So most thing is that stabilizes the double-stranded DNA is of course the base stack. The types of isomerization that occurs in the so, types of isomerization. Now there is something known as like keto-enol tautomerism. Shift a position of hydrogen acid either nitrogen or oxygen. Correct. So this is known as keto-enol tautomerism, which happens in the nitrogenous base, and of course the answer is tautomerism. And how it works is explained here. So there is a keto group here. What happens there if there is this is a polar, so this attacks the hydrogen here, this attacks the hydrogen here, and this hydrogen and go and binds there. This is known as keto-enol tautomer. So it's a ketone form. See the C double bond O ketone form. It is the enol form. Correct. So this that's how the keto-enol tautomerism works. The same happens also here also in so keto-enol tautomerism works from thymine and also in guanine. Now you can see when the ketone-enol tautomerism happens. This is the and there is also amino-imino tautomerism. Now thymine and guanine, both of them are mostly found in the keto form, right? So when there is thymine in the keto form, which is this one, you can say thymine is binding to adenine, correct? But when there is an enol form, now you can see this NEs have also this thing, and this has also now, and this also have some charge. So now as you can see, at the enol form, thymine can actually base pair with guanine, correct? So now this kind of anomalous base pairing actually leads to mutations in the DNA, which is a and there are separate mechanisms which will I think which I don't know if it will be taught in this course, but there are mechanisms so DNA repair mechanism that actually takes care of this kind of mismatch inside the DNA because T cannot bind with G in our system because now as you can think of like if you know about replication, I hope many of you know. Now if I have a sequence like this, so generally and GCSA So if I now, and the reverse sequence will be T A C C G T, right? Now, for example, this thymine was in keto form, and now this has become keto to enol form, right? So correct. This thymine has become an enol form. So at that time, then it will base pair with G, correct? If the G G C C G T, correct? Now, if you think of like when protein synthesis happen, if this and if this thing is stabilized at the next cycle of the DNA, if there is my coding strand, then at the next cycle of DNA, if this is my template strand TGC CGT, then at the next cycle it will be A C G G C. Now as you can see, uh, C A, yeah. As you can see, this ATG has now become ACG. So which is not correct, right? So now ATG codes for methionine. ACG will code for something else, right? And methionine is a start codon. So maybe the start it loses the start codon. So that's why this for this kind of uh, this kind of case there is a repair system in our body known as mismatch repair system or MMR. Mismatch repair. So mismatch repair take care of this kind of analom this kind of base anomaly inside our body. Okay. So anyway, this will be tautomerism. Okay. This has not been taught in the class though. Okay. The Chargaff's rule is not applicable to double-stranded DNA, double-stranded RNA, RNA hybrid, and single-stranded DNA. Now, what Chargaff's rules always said that A, so A will always base pair with T, and G will always base pair with C, and percentage of A will be always equal to percentage of T as well as percentage of G. Oops, it will be always equal to percentage of C, and in any species, so percentage of purines will be always equals to percentage of pyrimidine. Now this happens only if there is a double stranded structure, right? Because otherwise, if it is single stranded, so if it's single stranded like is in RNA, so A T G C G C A like that, right? So this doesn't matter. But when if you're doing a double stranded, it then only Chargaff's rule is applicable. So double stranded DNA, of course, Chargaff's rule will be applicable. Double stranded RNA, yes, again, even if there is a base pairing between RNA or RNA secondary structures like this, if it forms, then also RNA secondary structures will also have a form like this. RNA DNA hybrid always it's again a double stranded, but single stranded DNA will have single stranded structures. So my correct answer will be uh, single stranded. In Sangus method, chain angles stop at DD end tips because of there is some 2' hydroxyl group, absence of 2' 3' hydroxyl group, presence of 3' hydroxyl group. Okay, this you should tell. So because uh, this I want you guys to tell this question. I want to get this answer from you. Okay, because this has been clearly taught in the class properly. I'll give you 2 minutes. Let's see. Okay, still I'm not getting any answer from you. What will be the answer? Still no answer. Okay, I'll give you 1 more minute, and because then I'll give you the answer. So before that, this is the easy question down. What is the most dynamic RNA that undergoes turnover and acts as a template for translation? Guide RNA, not is no. I don't have to explain this because this will be mRNA. So mRNAs are like they will have a cap. This they will also have a poly A tail, and the ribosome will come and sit and translate the protein, right? Okay, very easy. Okay, okay. If no one is giving the answer, it's like absence of 3' hydroxyl group, and I don't need to, I think, explain this because sir has already very well explained how the Sanger sequencing work. Okay. Okay. So in carbohydrates, so carbohydrates are sugars. They are like the energy. They gives us energy to the most of the our in metabolic pathways, and they're used in our metabolic pathways to give rise the energy or ATP or GTP. So, sir has explained different things in carbohydrates, like what are different kinds of isoforms, epimers, everything. So I just briefly do a recap of what are D and L isoforms are, because this I feel this concept is very, you need to clear it very fast, and what are epimers. Okay. So I remember, oh, epimers or DL isoform, they're all stereoisomers. They're all of them are what is known as stereoisomers. So what are stereoisomers? Which change the angle of plane polarized light, correct? These all of them are known as stereoisomers, correct? Whatever change the angle of plane polarized, right, are known as stereoisomers. Now there are different kind of stereoisomers, but we are mainly interested of this D and L isoforms. Now D and L isoform, how do you differentiate between D and L isoform and other stereoisoform? Now, so D form, the sugar isoform D form is the, or its mirror image L form. Now how it is determined? Now if you think of a sugar like glucose have multiple chiral carbon, right? So most of its carbons are chiral. So like for example, this carbon is chiral, this carbon is also chiral, this is also chiral, this is also chiral, this is also chiral. Now how do you say that which one is um which one is the DL, which chiral carbon we should use for determining a DL isoform? Now there is the thing. So is determined by the relationship to the parent compound. Now all parent compound of the uh, parent compound of all the glucose are glyceraldehyde, correct? L or D glyceraldehyde. Now glyceraldehyde has a single chiral carbon. Now always remember, sorry, it has two chiral carbons, right? Now always, no, sorry, single, yeah, it will have two chiral carbon. Always remember, when you think of chirality, always consider this carbon C. Now these have multiple R groups. So R1 is our, here is CH, sorry. So R1 is our CHOH. There will be three more things. So CHOH. Now, if I consider only this carbon, okay, I'm considering this particular carbon. C. The other R group is H, OH, or CH2OH. Correct. Now, if I do a transition, if I put a mirror of this in this plane, correct. So if I just change it, so this, sorry, this will be CH, not CHOH. Sorry, my bad. CHO, correct. So CHO. If we change it, this will not change. This will be CHO only. So think of this carbon. This will shift. So now, if you consider the mirror image, so the CH2OH will come here. This OH will, if it was on the plane, it will go actually below the plane, something like that. And this will be H. Okay. Those will be always shift. So now, as you can see, this OH has been shifted to this side and this H has come to this side, correct? So other things we cannot, here we cannot say how this chirality is changing because it's above the plane, on the plane. Anyway, so you can see, think of like, if this is my plane, you can think these two are on the plane. This one may be below the plane, this and this one above the plane. When we change it, this comes above the plane and this goes below the plane. Something like that you can think. Okay. Now, if we think of, now whenever saying about D and L isoforms, always remember that the uh, orientation of H and OH group around the carbon atom adjusts to the terminal carbon atom. Now, what is the terminal carbon atom? So this is the, so remember, orientation of H and OH groups around the carbon atom adjusts the terminal carbon atom. This is my terminal carbon 6. So adjacent to the terminal carbon atom is this one, correct? Or the, or we can say also the asymmetric carbon or the chiral carbon that is farthest from the aldehyde group. So this is my aldehyde group. Now the last chiral carbon is this one because this is not chiral, right? This has two same R group. Two same R group cannot be chiral. So the farthest chiral carbon is this carbon, carbon number 5. So the H and OH orientation at the farthest carbon or the closest chiral carbon of the terminal carbon will determine if it is belongs to the D. Sorry, this will be capital. This will be capital D and capital L. D or L series. Now, if it is on the left side, it is L glucose. If it is on the right side, then D glucose. Okay. So like that. So when the sugar is D isomer, and it is on the left, and it is on the L isomer, most of the naturally occurring monosaccharides are D sugar, but there are a few we can also see. And epimers are little different. So epimers are not the same thing as D or L isomers. Okay, epimers are little different. Now you can, if you see this one, this image here, you can see this image here. So you can see that so epimers differ only the configuration around one carbon atom, except the last chiral carbon. Okay, this is known as epimers. For example, glucose and mannose. If you can see glucose, see this. If you can see C2, this is only separate. This is same. This is same. This is same. Okay. If you see galactose, this is same. This is same. This is same. Okay. HOH, OH, OH, HOH. At this you can see that C4 is changing. So D-glucose and D-mannose, D-glucose and D-mannose are only different stereochemistry as C2. So these are known as C2 epimers. Okay. And yeah, so this is the same thing has been also shown here, like how D-mannose or D-glucose are changing. If you see the aldose, there is also a hexacarbon aldose sugar. So if you see the aldose, it differs on two places: CH, OH, OH. So they are two stereo. D-glucose and D-aldose, they are stereoisomers, but they are not epimers. Okay. And so this, let's a small term is only applicable for glyceraldehyde. If um, if a glucose has only two stereoisomers, then they are known as enantiomers. So this is, for example, glyceraldehyde has only one chiral carbon atom, this one. So it has two glucose. So two only, like it has only two stereoisomers, so they are also known as enantiomers. Okay. Now what is anomers? Anomers is a different chirality than the linear sugars. So linear sugars doesn't have any anomers. The atom anomer comes only when we are forming a cyclic sugar or hemiacetal sugar. Correct? So this will either cause pyran ring or form a pyranose ring, or it will come a furan ring, or like fructose will call fructofuranose. Correct? Now how it happens, as you can see in this image, very clearly see there's this OH here and this OH here. So this extra electrons in this extra electron in this particular O. So this is a polar bond, right? So on OH there will be two electrons. So this electrons will attack the carbon here, and this carbon will be here, and this carbon will take the hydrogen from here. Correct? So that means then it will become a cyclic bond. So that is cycle. This can be either in this orientation, like where H is on the up, OH on the bottom, or in this one orientation where H is down, OH bottom. So these are two as alpha and beta. So remember, in nature you will always find in glucose, for example, you will always find alpha, beta nanomers as well as the linear glucose in a solution. at a same time. Okay. And yeah, so this is like, so there will be alpha D glucopyranose or beta D glucopyranose. And for similarly, if it's a ketose sugar like fructose, it will form a N-1 membered ring. Sorry, N-2 membered ring. If it's a ketose sugar, so it will form like fructofuranose. Okay, it will have 5-membered ring. And then we talked about how the disaccharides form. So as you can see, this is the 2 hemiacetal group in the alcohol. Now this will attack each other and will form condensation. Now there could be 2 types of bond. One will be the glycosidic linkage. So this is known as O glycosidic bonds, right? In maltose, you will always have alpha 1,4 because it's a alpha D, 2 alpha glucose are but yeah. So as you can see, one alpha D glucose and beta D glucose. So alpha 1,4. So the first carbon is alpha. This carbon number is 1, carbon number is 4. So alpha 1,4 glycoside. So for in maltose we will have alpha 1 to 4 glycosidic linkage, correct? Linkage. And in another case, for example, in case of this one, lactose, where you have glucose and galactose, and as you can see, they are C4 epimers, right? So C4 will be on the opposite side. So here you can see this is OH is down. Yeah, whatever. Like they have. One second. Yeah, glucose and galactose. So if you see here, yeah. So if you see here, there is a beta form. This is a beta D glucose. So as you can see, this is the HOH, the fourth carbon here is on the opposite side, and here the this carbon is H and OH. So this is departing here. Okay. So there H is down, here H is up. In this case, there will be beta 1,4 glycolizing because the first carbon is beta, so beta 1,4. So in case of lactose, that beta 1,4 glycosidic linkage. Okay. Then there are other examples also, like fructose. So glucose and fructose. So there will be like alpha glucose and beta fructose. So beta D, like fructose, beta 2 glucose, or it will say that glucose alpha 1 and 2 beta fructose, correct? So you can see here. So like that, alpha and beta anomers, depending on the which anomers you are using, alpha or beta, you will have like the glycosidic linkage will also change. And it's not necessarily will always have glycosidic linkage between the 1 or 4 or 1 or 2. You can all also have glycosidic linkages between this 3 or 4th carbon. So there are several examples. And yeah, so these are several. So this that so yeah, so monosaccharides. So monosaccharides, I'll just say monosac plus monosac. This is equal to disac and disac plus disac plus dot n number of disac will form a polysaccharide. Correct? Like that, we'll have like this. Okay. Okay. Now we'll come to the question. So yeah, so anything I am missing here, I have discussed everything here. Okay. The transporting sugar in insects and found in hemosyl and the walls of fungal spore. It's again remembering questions. I don't have anything to explain here. So this is three halos, correct? So the nitrogen containing polysaccharide found in cell walls of fungi and also in the exoskeleton of arthropods. It's a chitin and it's a polysaccharide, and you can see the structure of them. So I can ink annotations. Yeah, you can keep them. So I'll show you one chitin structure. Yeah. Get some easier image, very difficult image. Yeah. So you also this is also this, right? So chitin structure, yeah. So chitin is a polymers of a, this has a repeated unit of N-acetyl glucosamine. So as you can see here, so this is the N and this is the acetyl group. I copy this image. Yeah, I can copy this image. I could have pasted it, sorry, I could not paste it previously. Oh, I cannot paste it like. So this is a chitin molecule, and as you can see, it's a N chain of N-acetyl glucosamine. So N acetyl glucosamine. Okay, N-acetyl glucosamine in numbered of. So this, as you can see, this is a glucamine. So anyways, there is a amine group here. So that will then it will say it at glucosamine. Glucose plus amine. Amine is NH2, correct? Amine group. That will now tell you always that glucosamine. Now at this amine, there is acetyl group here, CH3OH. So CH3O, acetyl group is here. So that's why I'm calling it N-acetyl glucosamine. Okay. And so next, so this is chitin. The aldehyde group of one aldose reacts with a secondary hydroxyl group present in the same molecule, which will form, of course, aldehyde group in the aldose. So this is the reaction it's showing. The aldehyde group in the aldose reacts with the secondary hydroxyl group present in the same molecule, correct? This one, holding a hemiacetal formation. So this is known as, again, it is also written here, hemiacetal linkage. Okay, so it's a hemiacetal having a cyclic structure. Okay, and next is two anomeric form resulting from hemiacetal or hemicatical form. I just now explained it will be alpha and beta anomers. This one. So okay. So as I explained somewhere back here, okay, alpha and beta anomers. The only monosaccharide that lacks a asymmetric center. So glyceraldehyde, as also I have explained, and fructose and glucose, you know, they have a, so this is a L glucose, L glucose, and this glyceraldehyde. Fructose also is here. They will have lots of chiral centers like this. So let's draw diacetone. Now dihydroxyacetone. Okay. Now what is the structure of acetone? CH CH H H. Correct. This is the structure of acetone. Now dihydroxyacetone. So we need two hydroxy group. Oops. Two hydroxy group. One will be here. One will be here. So dihydroxyacetone. If I write in a simple formula, now is this carbon is chiral? This carbon? No, there is two H groups, so this cannot be chiral. This also cannot be chiral. So none of this carbon has a chiral center. So, right. So because there is two, so remember always to be a carbon to chiral, this carbon should always have three different R groups: R1, R2, R3. Correct. And C to be chiral, R1 not equals to R2, not equals to R3. This is kind of a thumb rule. Okay. If they are they any of them are uh, equal, then so in this case R1 and R2 and R3 are equal. In this case also R2 and R3 is equal. So this cannot be chiral. This anyway doesn't have any R3. Correct. This only have, and this R3 also, these two are difficult because this is also CH2H, this is also CH2H. So none of this carbon is chiral. So dielectric phosphate is my correct answer. The repeating unit of cellulose is known as cellobiose. So so these disaccharide, this is known as cellobiose. Okay. In I don't have anything. What do I say? I don't have anything to explain here as such, because as you can see, cellobiose is basically C1, 2, 3. So yeah, like two group. So the molecule is just three. Yeah. So I don't have to see names. These are all like remembering things. There's nothing to be explained here. So this is the cellobiose, this is the structural unit. I feel there's something like this problem with the THS session in this course, because if it is a mathematics related course or physics or something other where I actually can explain things in a little deep way or solve things, it will be, yeah. But here in many of the questions I don't have any explanation. These are all remembering things, like the part B in the CSR UGC net, where you have to just remember. If you remember, you have okay to go. Okay, and then we went about the glycolysis and the Krebs cycle in the course. So that the carbohydrate metabolism at the third class. Third. So whenever we are take up glucose from the diet, they kind of different pathways it can go. Okay. So one of the pathways of glycolysis. Then there could be glycogen synthesis as well as glycogenolysis. So this actually would be something like double-sided, but this is not been taught here. I don't think it will be. I don't know if it will be taught yet. Then there is this pentose phosphate pathway. This is important for NADP regeneration because this NADP is in the glucose. Sorry, oops, what is happening here? So yeah. So in this case, NADH or NAD or NADP, whatever, NAD is regenerated. NAD needs to be regenerated because from the cycles, next cycles, the the NAD is converted to NAD plus, NADH and NADH plus, H plus. So that needs to be regenerated. For that, pentose phosphate pathway is used, but again not been covered in this course. Then there is ribose phosphate pathway, which synthesizes to the DNA and RNA. It is another nucleotide biosynthesis pathway, which also has not been taught in this course. And this is a separate part. This is a separate biochemistry and metabolism course, which you need to take for this kind of this for explanation of these in greater detail. Uh, what else we have? Okay, and this so from glycolysis will form the pyruvate, and lactate is formed when there will be this when there will be um, sorry, anaerobic glycolysis. At that time, there will lactate will be formed, and otherwise, in presence of oxygen, it will go to the citric acid cycle, and also this pyruvate will be converted to acetyl CoA, and it will go and bind to the cholesterol. But can also form cholesterol and other different amino acid pathway, which is not been taught here. Uh, okay, this is a problem. So yeah. So in glycolysis pathway, we always have glucose here, and so from glucose is first converted to glucose 6-phosphate, which is known as the marking the glucose for the glycolysis or some other path. So the glucose is fixed in the cell. So fixation of glucose. But this particular step 1 is known as fixation of glucose. This step is known as fixation of glucose. Correct? Step 2 is conversion of from glucose 6-phosphate to a fructose 6-phosphate. Correct? So this one, fructose 6-phosphate to fructose 6 isomerase reaction. So now this is a first step. From one more phosphate group is added at the first curve. So fructose 1,6 bisphosphate. This is the first ATP. So there is a -1 ATP of generation, correct? Then fructose 6-phosphate to there will go to that will be divided in aldolase. It will be cut from between and it will form as a so it will basically cut from here. Mm. So it will have a cut from here. It will form the dihydroxyacetone phosphate, which is this one, and so this same as dioxyacetone. Now instead of OH is a phosphate and glycerylate 3-phosphate. Correct. So T different group are now formed. Okay, allulose and dihydroxyacetone phosphate. Then this there will be a isomerization reaction again by doing triphosphate isomerase, and then from there we'll have now two molecules. Now this is the steps. So we'll have one minus ATP here. Now from here we'll just generation of ATP. So this is NADH and NADPH. So then this will be converted to plus 3. So plus 3, basically plus 3 into 2. And in this case now, from this bisphosphoglycerate, so now glycerate is phosphate, and from glycerate is phosphate dehydrogenase, there will be bisphosphoglycerate, right? 2, 3 bisphosphoglycerate. So as you can, 1 3 bisphosphoglycerate. Right? One position, 3 third position. 1 3 bisphosphoglycerate. So there is a net ATP synthesis of plus 1 into 2, because always there will be two molecules of bisphosphate, plus then there will be 3 phosphoglycerate will be converted to phosphoglycerate, phosphoryl pyruvate, and phosphoryl pyruvate to pyruvate again converted done as pyruvate kinase. So this will be also plus 1 into 2. So the net ATP synthesis of this is when in presence of plus O2 condition, you will have 6 plus 2 plus 2 minus 2, so is equal to 8. At minus O2, when there will be anaerobic glycolysis, and this 6 will be not there, so 2 plus 2 minus 2 is equal to only 2 molecule of ATP from glycolysis. Now this pyruvate will now go to instead, so this pyruvate, this step is not shown here, I did not show here. So from pyruvate to pyruvate dehydrogenase enzyme will come here. Dehydrogenase come, and this will also cause one NAD plus two, NAD plus 2, NADH plus H plus, to give rise to a acetyl CoA, correct? Acetyl CoA. So now this acetyl CoA will go to the Krebs cycle phi. So this is the TCA cycle or Krebs cycle. Okay, now there is always the 3 carbon intermediates, correct? So then it will be a citrate synthesis. So there here will be plus 3 ATP. Then here citrate synthesis, then citrate to cis aconitate to isocitrate, then isocitrate dehydrogenase. So there will be plus 3 ATP again. Then from isocitrate citrate to oxaloacetate. At this step again, plus 3 ATP will go. Sorry. From isocitrate, from succinyl C with alpha ketoglutarate, it will go to succinate. And from from succinyl CoA, the succinyl CoA to succinate, it will again have a ATP synthesis. So there will be plus 1, and then there will be from succinate to fumarate, there will be a succinate dehydrogenase, which will to use ATP, but the difference is that it will have only 2 ATP in the ETC electron transport chain. And after that, succinate to fumarate to malate. Again, malate dehydrogenase step, there will be another 3. So total amount of, so here I am always getting plus 3 here. So 3 plus 3 plus 3 plus 1, 10 plus 12 plus 3. So from one molecule pyruvate, I am getting 15 ATP molecule, correct, in presence of oxygen. And a Krebs cycle will only happen in presence of Yeah, in presence of oxygen, right? So in this step only, so in then respiration where we are giving CO2, here we are giving CO2. And remember, glycolysis happens in the cytoplasm, whereas this happens in the mitochondrial stroma or mitochondrial inner part of the mitochondrial. So mitochondrial stroma, correct? This part. So one molecular pyruvate generates 15 molecular ATP. Since we will have two molecular pyruvate, so two molecule will give 30 ATP. So one molecule of glucose at presence of oxygen. So 8 plus 30 is equal to 38 ATP. So one molecule of glucose will give me 38 ATP total. Okay. And yeah, so then we have also discussed like Krebs cycle in other pathways. So in Krebs cycle intermediates can go to different pathways, and I'm not going into much, I'll not go to much details of this part because this was again discussed quite well in the class, and it's not also like it was discussed in the class and it was not discussed in quite like very detail, and this not required. So always remember, so there will be a path this is, so from Krebs cycle intermediates we can get glucose, and this step is known as neoglucogenesis. I hope I could have taught you. I'll can inform like maybe in the next class I can give you a one slide. I'll show you one slide about neoglucogenesis, how it works. But this can be, so neoglucogenesis is basically reverse of the glycolysis. So and this is something I'd be very happy to explain because recently there was a paper in Science. Let me see it. Recently there was a paper in Science and which has actually shown something very interesting about neoglucogenesis in uh yeah in hummingbirds. So hummingbirds, yeah. So hummingbirds are Um, so the paper title was Loss of the Glucogenic Muscle Contributed to Adaptive Metabolic State. Now hummingbirds can see they have this hovering flight. Now this is like 1 by 30 seconds. If you see this kind of hovering flight in hummingbirds, now they flap these wings around 80 to 80 times. Okay, rapid wing 80 to 70 to 80 times a second they are beating the wing. So this is very slow motion image. Let's see. So for this kind of wing flapping or wing beating, hummingbirds have like very high metabolic rate. Now to do that, hummingbirds mainly depend on the glucose. They don't like glucose or fructose, correct? Because now they feed on nectar. Nectars are mainly are glucose and fructose. Now to maintain this kind of hovering flight, what they have shown is that they have increased glucose transporters and all. Thus you can understand there is increased glucose transporters and all. But then they go off and show that, so as you can see, this kind of hovering flight. See, this is the hovering flight. Okay, this is a hummingbird. This is a long-tailed hermit, and you can see how it is taking the nectar. So ultimately, these people and gone and show that. So this is the thing I wanted to explain. Now, gluconeogenesis means you are always glowing on going back. Okay, you are glowing back. So from pyruvate to glucose, you are going back. So this is glycolysis where you are getting glucose 6 phosphate to pyruvate, or you are going back. Now going back, only a few steps needs to be changed. So as you can see, most of the steps in this pathway are uh, bidirectional, right? Most of the pathways. However, some of the pathways are single directional. For example, the fixation of glucose. Once the glucose is fixed, this pathway, correct? Once the glucose is fixed, it cannot go back to make like glucose 6 phosphate to again glucose. Right? This path cannot be reverse oriented. Again, another path where the ATP is required. So whichever steps that the ATP is required, those steps are always either ATP formation or ATP degradation. Those steps are always single sided. For example, this phosphofructokinase 2 fructose bis fructose 6 fructose 1,6 bisphosphate. Then there is other path, and then there are other another pathway where ATP is generated in case of glycolysis from phosphoenol pyruvate to pyruvate. There is a pyruvate kinase. So these steps are uh, monodirectional, not bi. Other steps you can always see there is a bidirectional step. So enzyme can both work on the single side and opposite, because chemical reactions are mostly bidirectional, right? So A plus B can always from A plus B can always from C and D, but always C also C can D, C and D also can form A plus B, and these are like chemical, these are like very common concepts in the chemical kinetics, and people may have learned that in the elemental chemistry, the dissociation constants and association constants like that. Okay. Anyway, so in gluconeogenesis, to bypass this kind of single like monodirectional pathway, so there are from pyruvate. First, we using two molecules of ATP, pyruvate is converted to oxaloacetate, and using pyruvate carboxylase, and in using paper carboxylase, using another two molecule of ATP, so two molecule GTP, it is formed phosphoenol pyruvate. Now again, this part is fully bidirectional. So total we am use I okay. So I am using, so for gluconeogenesis I am using 4 ATP, like minus 4 ATP, among them are like minus 4 actually ATP, so there will be minus 2 ATP and minus 2 GTP for our reaction. So I am losing two, total 4 molecules, 4 energy molecules. Okay. Now as you can see, if it is converted, then at this step, instead of there is one enzyme called as fructose 1,6 bisphosphatase is used, and as well as glucose 1,6 phosphatase. There is a kinase and there is a phosphatase. So kinase adds phosphate and phosphatase, sorry, removes phosphate, right? And so these two cannot work together. There should be some control which one should work. So that's why here is the fructose 1,6 bisphosphate and glucose 6. Now these people have shown this hummingbird actually have loosened this fructose 1,6 bisphosphate enzyme, because they don't need gluconeogenesis, or whatever glucose is taken up, they wanted to put everything to the Krebs cycle. They don't want any kind of gluconeogenesis. So that's why they use, and yeah, so anyway, BP, and they have shown in different experiments that the gluconeogenic muscle enzyme BP2 was inactivated in the ancestral hummingbirds during the evolution, because hummingbirds doesn't need gluconeogenesis. Okay. So this was the glucone. Basically, I have just explained you the gluconeogenesis. So remember, the gluconeogenesis, there are three enzymes that are separate. Sorry, four enzymes that are separate. Okay. Pyruvate pyruvate carboxylase, pep carboxykinase, fructose 1,6 bisphosphatase, and glucose 6 phosphate. I'll put this image on the other slide also. But remember, this four are the uh, enzymes that are responsible in the gluconeogenesis. Sorry, these four enzymes are required for the gluconeogenesis, and this is a kind of a quite important thing, correct? What is happening? Okay. So the example questions is glycolysis, phosphonomer pyruvate is converted into pyruvate with help of pyruvate kinase. I recently explained the whole glycolysis pathway here. It is of course pyruvate kinase. The net balance of ATP, the net balance of ATP produced for oxidation of one pyruvate molecule in Krebs cycle. One pyruvate molecule in Krebs cycle. Okay. Now, if you don't consider ETC only in Krebs cycle, then only one ATP is forming in place of GTP. But if you consider that ETC is happening, as I've explained here, so if you go one molecule of pyruvate generates 15 molecules of ATP, correct? So 15. I have to explain the Krebs cycle, to explain in mitochondrial storm. Okay. Lastly, we have talked in the last class, we had in the week 3, we had talked about lipids. So yeah. Sorry. So lipids are different types, and I will not go in details of how the lipids what they do because it has been again explained in the class. So there is one thing I think it is not explained in the class. So there is a naming conventions in the fatty acid. First, naming convention. If there is a unsaturated fatty acid, then all you write. So it's a saturated fatty acid. You say just write. Mm, eight. Say this is if there's a saturated fatty acid, I have to write 18 octa deca octadecanoic acid, correct? Anoic, octadecanoic acid. Since there is a two, so there is a one double bond here. Sorry. So what will I write? There is a one double bond. So 18 in this after colon I'll have the number of unsaturations. Okay, I'll write number of unsaturations, and then I'll write okay at which position. Now where at which position the delta is at the C9 position. Then since it's a octa deca, it's a enoic acid, correct? Because correct. So I will write like this octadecane. So in methane, like methane, or like say butane is 4, which is any unsaturation. Butene is 1 unsaturation, like that. Okay, like said octadecaenoic acid. And if you have multiple double standard form like this, so 1 2 3 4 5 6 7 8 9 10, so then you will see that okay this is the all the position. Now another example is that here the carbon numbering is started at the, so here you are going to see the carbon numbering is started at this carbon. Okay, but what if I started the carbon numbering at this position, which is done here? So that is known as omega. So as you know that omega-3 fatty acid is very like it prevents heart disease and all. There's lots of advertise that it's in the oil advertise you can see they will say that omega-3 fatty acid. So omega-3 fatty acid means omega position is from the right side position of the fatty acid. So you can see omega-3 fatty acid means it's as a uh, it's at the unsaturation at 3 position. So that's why it is the omega position. So omega is going for the from the at the end of the chain, right? So the alternative convention numbers of the carbons in the opposite direction assign the number 1 to the methyl carbon at the other end of the chain, and this is also known as omega. Correct? And there are like zigzag representation and representation and so. So there is a cis. So now there is also cis, and if you have a trans, so trans would look like this. Okay, the double bond would look like this. Then if you like cis 9, then cis 9 and trans at this position like that we would have even explained. Okay, and there are different like different separation of lipids. So storage lipids which are neutral is a glycerol, and we'll have fatty acid for triglycerols. So one of the triglycerols like LDL, VLDLs, those all lipoproteins kind of, but all of those are triglycerols, correct? Triglycerides and all. So yeah, so that's triglycerol. So glycerol with fatty acid chains. There are glycerophospholipids which have like glycerol, fatty acid, and then it will also have a phosphate and alcohol. Then there is sphingolipids which have like sphingosine as its like uh instead of glycerol it will have sphingosine at its anchoring molecule, and you can see there will be sphingolipids, um, and phospholipids and glycolipids both, and sphingolipids can be both like phospholipids or either glycolipids. So if it has a fatty acid as a oligosaccharide chains, then yeah, then sulfolipids and all is also there. Then archaeal lipids which has like some different concentric different types of archaeal lipids, so will not which has not been taught in the class. So there's like a different types of lipids. Okay, and we'll also have cholesterol. I have not discussed here. Cholesterol I think has been discussed in the class. Okay, so the first example question is that um, the low molecular soluble lipids that can be transported from the blood plasma without transporting protein is ketone bodies. This again you have to remember, there is no one thing to explain. So molecule that is involved in aggregation of platelets. Now one of the platelets you are like thrombus mean, so thrombus means clot clots. So thromboxane, Neilman Pigrising, and there was a lots of like this was a list, and then this is also nothing to explain. This is pingomyelinase. Okay, and yeah, so this is the like these are all the questions that has been there in the keep all the previous year's assignment, and there are two more questions. So one was that the glycerol biosynthesis was not been taught in the class, but there is kind of a pathway you can just have a look. And this the immediate precursor of cholesterol is of course 7-dehydrocholesterol. There are two questions I don't know. It was not been taught in the class here, and protein catabolism I think has not been taught yet, so I'll not explain this one. Okay. So yeah, so these are all the things that I had to talk about in this third session of the third TA session. So I hope it has like it has helped you understanding the concept a little more better. And please feel free to contact me because I have again I am just repeatedly saying I have you can always contact me as this email ID shobikshom@isc.in. If you have any doubt or you want to have anything special, such like anything want to be discussed in the class, like you want this to be discussed, if it is feasible, if you can mail me the topic you wanted me to discuss in the class, and yeah, and if it's feasible I'll surely discuss in the class. So that's it, and I'll just again ask, so if anybody have any doubt. You missed two assignment. Is that okay, sir? Uh, I mean, if you will miss assignment, you will. I think they consider best of. I think if I am not sure because these kind of things has been not discussed with us. So we are mainly the TAs of the course. So assignments and all, I don't know if you have missed and all, if it will get a problem. You can mail the NPTEL team. Okay. I am not sure that if how much it will affect you. It will somehow affect, but I'm not sure. Then mail to the it will affect your grade, I think, but how much I don't know. Anything else, anyone? Okay. Then if no one is there, let's end the meeting here. Thank you everyone for joining, and we'll meet next Tuesday. Okay, on 28th. Please paste your email. Okay. Uh, okay, sure. I can do that. I think previously also I've done that. So this is my mail ID. And okay, thank you. Thank you a lot.